I'm ready. Well, thanks for attending the DNA seminar. And what I'm going to try to do is give you a little bit of background in how we do DNA, some case examples that I hope you'll find interesting of wrongful convictions and how I was able to be a part of undoing them. And mostly what I want you to do is get a mindset of how do we change things to make them better. And I'll say that the lawyers who go to battle for defendants, I respect in the system because they protect us from the overreach of zealous prosecution that the state is interested in to make sure that bad people are kept out of society. Those battles are fought every day. And when you win a battle against bad science for us, you don't just save your clients. When you launch an objection about bad science, you don't just affect your case. And especially as you win some of those challenges to bad science, you protect all of us. And I am very grateful. Our rights mean nothing unless there are those willing to fight for them. So thank you for your work. And I hope to teach you well. I hope you can help us to change things that are wrong in the system. So I want you to keep your eye out for improvements that you can make. So we have 3 goals for this. What was I was hoping would be a discussion. You're unfortunately going to have to listen to me. Some of the changes that labs are making and why they're making them. And your cases will probably reflect some of those over the next few years, but also you'll be handling cases where these changes have not been made yet. Things that we as a system can consider adopting and things that you can consider in your practice being effective against bad practices, introducing good practices, and opportunities for improvement. So I really want to focus on like how we can move the ball from errors that we recognize, which is the first step of any of this. You know, being a hypocrite is the first step of any revelation of any improvement in a system or person. And so I want us to keep that in mind, that we're looking for problems that we can fix. The educational goals, I want to learn from mistakes that I have seen and some that I have made. Review wrongful conviction examples, examine common challenges to analysis that you can deal with in your casework, and the brainstorming part, you're just going to have to email me. This is first case we'll talk about. This is Terry Tolley, exonerated by brilliant lawyering and patient work from the Georgia Innocence Project, where I I'm still an emeritus member, helping them with DNA, and I have a current grant with them. So in this case, I was asked by the director of that project maybe 12 years ago to look at a rape case where a man was convicted and said he was innocent. So my work really wasn't that difficult in this case. There was some mitochondrial DNA evidence, some hair evidence. Broken hair used to be we could only use mitochondrial DNA. Now we can actually get full profiles from broken pieces of hair, just as of this last summer. But in this case, it was mitochondrial DNA. We'll talk about what that is. It was very clear that Mr. Tolley was excluded, and I was hoping he would get a new trial. When I talked with the lawyers, though, they didn't seem to be overwhelmed by this news. They seemed like they expected a much longer battle than this small one. And that was because there were 4 different convictions, 4 different rapes, one of which he pled guilty to. So how can this happen? Well, you know, I mean, as a scientist, I just think, you know, error times error times error times error. It should be a very small number of cases that would fit this. I will tell you that many lawyers who handled Mr. Tolley's case over the years thought he was guilty, certainly the prosecutors. And I like to remind us that we're all the same human. It's not like there's prosecutor DNA and defense lawyer DNA. We're all subject to the same types of biases. So we have to be aware that you and I will handle clients, cases where people claim they're innocent and we will think they're guilty and we will be wrong. That will happen sometimes. Mr. Tolley is innocent. The new prosecutor set up a conviction integrity unit, and they were able to go through this case carefully and undo all 4 convictions, thanks to the dedication of people who believed in challenging the system. Okay. I like to talk about what DNA can do, first of all, to show you the great things. So certainly Mr. Tolley's case is an example of what DNA can start to do. And this is another Georgia case that I worked on. John White in the middle here. The only reason he contacted the Georgia Innocence Project is because I believe Amy Maxwell was the director at the time. We had interns from law school. I was teaching them DNA. There were no good cases for them to work on. And so Amy decided that she would write all of the rapists and murderers, convicted people in the Georgia system and ask them if they were innocent. And John White, who had never reached out to the project before, wrote to the project and said he was innocent. There was a very dedicated law student on this case, and we were able to secure some DNA evidence. It was a 74-year-old rape victim. She was beaten as well as raped. When she was well enough, they showed her a photographic lineup. John White was the only person who was in 2 lineups. She was shown 2 lineups, and she picked him out in the second one. She didn't pick him out in the first one. You can imagine that the neurons that fire, the kind of recognition signals that fire when we've seen something before, are very hard for us to distinguish in terms of when we saw them before. Think of when you see a movie actor in a movie and you're like, where did I see that actor before? And people will have different opinions, and eventually you might suss it out or go to IMDb. But our brains get fooled very easily about recognition when they're subjected to this kind of subjective situation. And so anyway, the victim picked him out, and John's standing right in the middle. You might be wondering, he looks kind of like, boom, look at me. And that's what it was. They asked him in this lineup, where do you want to stand, John? And he's like, let's get this over with. I want her to get a good look at me. And there he is. And she picked him right out. Now, the curious thing about this, and what I like to remember from this case most, is that the law student, when we processed the DNA at the Georgia Bureau of Investigation, they said they got a hit to CODIS, to a convicted offender, with the DNA from the rape kit of the 74-year-old victim. And it was somebody who was in the system. Well, the student who worked on this said, I know that name, James Edward Parham. And we said, well, he was in the system. A lot of people write to us. Maybe he wrote to us. You saw the letter. You might have reviewed it at some point. No, it was in the John White case. I'm like, well, good luck. And so he went through all the records again and found out that on the back of this photograph, James Edward Parham's name was written, and there he is. He's the guy who matched the DNA. So, here we have the effects of bad subjective criteria, and then the DNA science, which is less subjective. It's very easy to pick out matches. But even in something like this, what can go wrong? You're good at doing that. Defense lawyers are good at trying to figure out what went wrong. And you might be thinking, well, maybe the DNA is wrong. And sometimes it can be. In this case, I don't think it was. But that kind of careful scrutiny, making sure that everything adds up, that you have corroborating evidence for what you believe is very important. This is another Georgia case. It was the first one that the Georgia Innocence Project had. The director had written the law, the DNA testing law. This was our first test of it. We drove down to southern Georgia, spent the night there. I swabbed the man in prison. I told him, look, if it matches you, everybody's going to know. It's going to be on the front page of the paper in this little town. And in fact, the DNA evidence confirmed guilt. Now, once again, it's just DNA evidence. Does that mean that tells the whole story? No, but there was a trial that had already convicted him, so this certainly did not help him. And I like to have this because it helps me remember that, you know, someone told me God will bless me forever. And I know that that can't be refuted by science, not in my view. But there are some claims that can be refuted by science, and that's what I'd like us to consider. So what is science? How can we apply it in our daily lives and in our casework analysis? Well, I got to work on Amanda Knox's case, a young woman who lived in the state next to us in Washington, and was overseas in Italy, and was tried and eventually convicted of killing her roommate. And when I started on that case, she was already at trial, and I was helping as like an international expert, showing the Italian findings in light of international standards. Anyway, we lost the trial. You may know the story, and there's been plenty of movies. But at about the same time, I was trying to get my slides together for teaching this subject. And I went to a book that I had gotten from my father's library. My father was a physicist. I don't think he initially considered biology a science. Eventually he did. And physicists have books with just a lot of numbers. And it turns out he had a great study filled with these books. But there were some books by scientists, and this was Galileo's biography. And so these are some quotes from Galileo. By denying scientific principles, one may maintain any paradox. The paradox of Santa Claus sliding down the chimney, the great deception we teach our children every year in order to train them that society can lie to you, even your parents. There are reasons why. And you need to be very careful about what you trust. So apply this technique. We're basically teaching children, yeah, it did bother you that a fat guy could slide down a chimney hole. And what about people with no chimneys? And you know what? That was okay because it wasn't science. But when you want to use the tools of science, you have to resolve the paradoxes, the conflicting information, or you have to have a recognition of that conflict and discuss how it might be resolved. In questions of science, this is, I think, so important to teach everybody, especially those who are going through the logic of a case and the science of the case. In questions of science, your humble reasoning, what does that mean? You got to look at the documents. You got to follow the trail of the samples through the case. You don't rely on your experts. You question your experts. You have a relationship with your experts that allows you to question them, not endlessly pester, though that's allowed. But you need to make sure that it makes sense to you, because if it doesn't make sense to you, it might be wrong. One of two things: if the science doesn't make sense, you haven't studied the foundational principles of that science, and you might want to get to know those. And I know lawyers do, they cram for that stuff before trial. Or in fact, the findings even of the experts are wrong. And I've been taught things by lawyers who just reasoned through a case. And I was like, oh, I missed that. Count what is countable, measure what is measurable, and what is not measurable, make measurable. So that principle is what we're trying to do. So if you see in a scientific report something that is an opinion and there's no measurement, something like, we tested for semen and there was weak positive. What is a weak positive? That's not a number. Is that like 0.01? Is it 10? Is it 50? Is it 99%? What does weak positive mean? Well, we have some idea, but you can see how that could be misconstrued. Can you be, telling the truth weakly? And science proceeds more, this is Galileo, by what it is learned to ignore than by what it takes into account. I know not to buy DNA shampoo because I know, having studied keratin for a year as a technician in dermatology at Yale, and having studied DNA for the last 30 years, I know there's not a whole lot of interlap between those two compounds. So I don't buy DNA shampoo. There he is, Galileo. Okay, when I started in this, I was a fruit fly biologist. And then I went to Australia. I got my PhD in a fruit fly study on DNA. I went to Australia and studied mammalian sex determination, because those wonderful hopping creatures, the marsupials, and the even more interesting platypus and Spiny echidna that lay eggs, these mammals were very different than us as mammals, but we were all looking for, in our little group of scientists, the small region on the Y chromosome that causes a mammal during development to grow testicles. That was seen as kind of the key to sexual dimorphism in our lineage. And so in studying male DNA and how you can look at very small pieces of male DNA, I found that I had a tool that my friends who went into forensic science would like to use. And so that's how I got roped in about 1990 into forensic science, a little bit, but I was mostly just a regular molecular biologist. When I went into forensic science, I was teaching in a small college, and my students, if you were live, this would be true. My students only get credit if they go to a talk, if they ask a question. Twice as much credit if the speaker cries. And that's because error is how science proceeds. My students have to find what I'm ignorant or wrong about in order to get a PhD, what everybody in the world is ignorant or wrong about. And so error is a really good place to look to make a contribution. So when I went to my first forensic meeting, I asked the head of the DNA unit of the FBI, who was giving a talk to 200, 300 analysts. I was in the back with a couple of students. And, you know, I always make them ask questions. I had to ask a question. So I raised my hand kind of timidly and got picked on eventually from the back of the room, and all the heads turned back. And I thought I was just lobbing a softball, but I was doing my job. And I said, what is the error rate of the DNA lab at the FBI? And guess what he said? 0. And in one of those moments of spontaneous comedy, I said out loud, where did you get such a small ruler? Because, you know, to measure 0 as a scientist, that's precision. So, you know, I wanted to know what they did. And he really didn't have an answer and kind of dismissed me. But I realized I was in a field that was not examining error. So I came up with this following teaching lesson, which is, if we measure the justice system by successes, all right, if you measure your practice by successes, your relationship, your struggle with whatever, weight, drugs, alcohol, if you measure it by your successes, you are unlikely to discover what you need to improve. You are unlikely to actually discover your hypocrisy and make the improvements. So the way I set this up, I had been to Salem to give a talk at a scientific meeting there, and took the tour of all the witch things. And I said, well, if we measure the justice system by the number of truly convicted people, then the Salem witch trials were an astounding success, because more than 100% of the witches were convicted. So that's, again, this idea that we make 0 mistakes. That's because you're only measuring your successes. So we studied the first 194 DNA exonerations. This is back in 2010. Now there's a gigantic database you can look at of the exonerations, the exoneree database, and find out about their cases, search using some tools. But this was useful because I would be receiving letters from prisoners, and we'd try to get testing done on a cigarette butt. And a judge would say to us, well, who's ever been freed by a cigarette butt? And I would say, well, Cigarette butts are 3% of the cases we've studied, Your Honor. And somebody might say, well, who's been freed from fingernail evidence? 5%, Your Honor, of the 194. So this concept of doing the science is because this research is the biography of science. And so it's important to study that. And you can, Google or Google Scholar is a great way to study anything that's in your case. Transfer of DNA, does DNA survive in the laundry? All of this stuff has been studied in peer-reviewed journals and published. That doesn't mean it's the last word on it, but it's a lot better than some expert who has never done an experiment in their life telling you about what their opinion of a sample is. This question of validation and documentation, you need to get the standard operating procedures of the lab, and you need to look at the validation summary. If you're doing an alcohol case, the alcohol people are great at this. They look at how the machines are calibrated, et cetera. But in DNA, there's also standard operating procedures and validation. How low of an amount of DNA does that lab have evidence, validation that says they can look at? Because some of your cases are going to have very small amounts of DNA with very big conclusions. Are they legitimate? This was a case, the only case I've done in mainland China. I didn't have to travel there, but basically there were 5 people who were convicted of killing a woman, hacking her to pieces, and disposing of her body. And the evidence was some mitochondrial DNA taken from a sink in the family's home. We asked for the evidence, the data, and the validation, not just the report that says it was a match. What about the data? Where are your findings? And I was a little suspicious. I'd never done a case in China. I didn't, I had all of that xenophobia that you have when you're first doing something new in a foreign place. But the judge there was very careful and allowed us to request the documentation, and there was none. And the judge ordered a new trial and freed them from custody. And so this is a foundational principle that you need to follow up on in your cases. Always ask for the standard operating procedures, always ask for the validation summary. I have a standard discovery order. I'm happy to share it with you for free. In mine, I keep updating it. Other DNA experts ask for slightly different things, but we're all asking for similar things now, I think. So how does DNA fingerprinting work? Here's a cell. You've seen them. Some of you know them quite well. And there's a nucleus. And here's a mitochondrion, this bean-looking thing. They're actually much smaller than this. You can have 1,000 or more in a cell. We get them from our mother in the egg, not so much from our dad in the sperm. Maybe rarely one gets through, but really it's all from mom. And that mitochondrial DNA was useful, again, in broken hair fragments, very degraded bodies. But I think we're not going to be seeing much of that anymore because there are new techniques that look at very small pieces of DNA. It's basically the genealogy techniques that are far superior than getting mitochondria. How do we get them? There's a swab. You can do it with a Q-tip. You don't need a fancy swab. Here's a Q-tip. DNA, that's it. I want to plant this on a weapon, but just rub it on the weapon. No, we don't do that. I want to analyze in the lab. I just take it and put it in a little tiny test tube. And let's see, do I have, just to show you the little tiny test tubes, this is what I mean. Here's a strip of them. So I'm just going to put a piece of this, cut it off with a razor blade and put it in there. And this is going to feature prominently in my talk because we work with tubes this size and then trays that look like this that can hold 96 of either these tubes, or we just put the liquid in them and they look like this. This is actually a holder for the tips, but that's exactly what they appear to be. Okay. And the cells that we chiefly work with are these kind of epithelial cells. Where do you get them? Your hand, any of your mucous membranes, mouth, anus, vagina, penis. They all look kind of like this. There are different kinds of cells that are sperm cells that I'll show you later. and then there are blood cells. But really, epithelial cells and the difference between epithelial cells and sperm cells is going to figure prominently because of sexual assault, because most of the accused are males, many of whom have left semen. Semen and ejaculate has about 300 million sperm cells. So we're going to look for those in a swab that might have a lot of these cells that you're seeing here, epithelial cells that are in the mouth, the vagina, the anus, the penis, etc. Okay, so how do we get this DNA out? Remember, there's lots of mitochondria in there, but mostly we're going to go for this nuclear DNA. That's what goes up to the database. The national database is pieces of the nuclear DNA, and all we're going to do is measure. We're just going to measure how long a piece you got at a particular site on one of your chromosomes, how long a piece you got from your mom, how long a piece you got from your dad. They might be the same size. You might get two of the same size. We look at that at 13 areas, just how long they are. We call those alleles. We get 13 areas, mom and dad, so up to 26 at most, and that's enough to identify you. And we do it through a technique where, if you could see the chromosomes, I can in a high school microscope, you can see the chromosomes in the cell. You probably can't get down to this level. This is a higher magnification here where you see the curly DNA. That would be an electron microscope. And if we could see them, and some of the scopes are starting to give you these shadowy pictures, you could see the individual bases in the DNA strand. But all we care about, I can't emphasize this enough, in the current technology, not the genealogy, but the crime labs, all we care about is the length measured by a process called polymerase chain reaction that copies and copies and copies between two little primers. We just care about how long certain pieces are. Not the 3 billion bases. I don't care about your AGCTs in the crime lab. I care about how long a piece of AGCTs you have at a particular area. That's made up of a pattern of repeating, usually 4 bases, AATT or TTGC or whatever it is. How many of those repeats, how long it is. That's a short tandem repeat. We abbreviate them here, STR, which is short tandem repeat. We call the locations loci or locus for individual ones. So chromosomes are fun. These are your chromosomes. If you have amniocentesis, you might have seen these. And the big one is called number 1. And they tried to name them before they knew totally what they did by size. They did make a mistake at one level, but that's okay. And then we have the X and Y. I'm showing you both of the sex chromosomes. For this person, what is it, male or female? Male, right? Y chromosome tells us male. Okay, so looking at just those 13 regions, the ones originally required by the FBI to load up to the combined DNA indexing software, which loads it to the national DNA indexing system in this, those 13 are enough to sufficiently discriminate you from anyone but your identical sibling, twin, triplet, etc. So what do they actually look like? Well, in order to explain this once to a judge, I brought in my, at that time, 4-year-old's Legos, and we're not looking at all the DNA. So if I took the 4 bases of DNA, AGCT, and I turned them into 4 colors of Legos, here is a pair of alleles, small section of the chromosome. Let's say it's chromosome number 3 from both parents, and I have one from mom, one from dad. And the question is, what's the difference? Well, the patterns look very similar in the flanking regions, but in this middle region, the short tandem repeat, in this case, 2 bases, AG, 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 or yellow, white, yellow, white, yellow, white. In that region, the parents differ. This allows us to do identity. We will call the allele from the chromosome on the left a 3 because it has 3 short tandem repeats, STRs, and we'll call the one on the right a 5. Now there are partial repeats, like if we only had an extra yellow on one of them, we might call it 5.1. And the repeats we use in in forensics are actually usually 4 bases, or some of them 5 bases. But here I'm showing you a 2-base repeat just to show you we're not sequencing every base. We're not looking at every chromosome. We're looking at 13 or 23 locations with the new kits. All we care about is how long they are. And we're going to determine that by starting at a part that is the same in everyone, the flanking regions, where we have a little primer to start the copying of PCR that goes to another primer at the other end where we are the same as everybody else. And because of that, we can use the same primers for every human, and it just copies back and forth, and we get 2 different sizes. Now, as I said, both parents may give you the same size. They might both be a 3.3 or a 5.5, 5.1, 5.1. But this one is 2 different size. We call that a heterozygote instead of a homozygote, the zygote being the egg and sperm coming together. Great, so we're concerned with the number of short tandem repeats. That's what your crime labs do. So what are the steps in all of this? Well, the first one is you identify a stain or an area to test if it's not clear. And you can do that with an alternate light source, basically a black light and some goggles that help protect your eyes and block out certain colors. And that identification of stains evolved out of blood typing. And so we still refer to it as serology, the study of serum in blood, serology, but really it's the identification of types of fluids now. And there are chemical tests that you can do with serology, looking at the different proteins in semen versus blood versus maybe urine, saliva. Then we have to move that DNA to a tube of water. We use usually a swab, but there's also this MVAC, invented in Idaho, that it's like a wet dry vac. You actually hold it like a wet dry vac, and you can go on a couch or a piece of underwear or a rock, and it shoots water and collects it back and puts it on a filter. So it's like a swab, but in a vacuum cleaner. Or you can cut and soak, as they do with cloth, and also there's a soaking method for spent shells. Now there are several groups claiming they can get DNA even after that tremendous heat that happens in a gun. They're getting DNA in some cases from spent shells. Then we have to get the DNA out of the cells in that tube. How do we do that? We got to break open those little cornflaky looking cells I showed you, the epithelial cells. Do that with a little bit of detergent. That doesn't break the sperm cells though, which is nice because then we can try to take all that broken cell stuff out with its DNA from the epithelial, spin down the sperm cells, and now I have a fraction that I hope is enriched for sperm cells. There's some problems with that we'll talk about. That's called differential extraction, to get the sperm cells away from everything else, wash them off, and find out who, one or more males, left sperm. After we extract the DNA, we have to tell how much we have. And so we take our little tube, we take a little out and put it into a PCR reaction, a polymerase chain reaction that just looks at a few different sites, tells us how much total DNA, how much male DNA, and then how degraded the sample is, how small, does it work well with small pieces, large pieces of DNA. As DNA degrades, the large pieces get cut, they won't amplify, we won't detect them. And so if we get more small pieces in the quant working than large pieces, we know that DNA is degraded. So in the quant, the newest kits tell us total amount of DNA, or concentration of DNA in the liquid, the amount of male DNA, and then how broken up the DNA is. Does it work for big pieces, in which case the DNA is not broken very much? Does it work for small pieces? And what's the difference between the efficiency of the big and the small? Then we want to copy not just a few pieces to determine the quantification and the degradation, but now we want to do a profile. And that's going to be, in the new kits, 23 locations. We're going to want to know how big mom gave you, how big dad gave you. Sometimes dad's small and mom's bigger. And we do that at least 13, but often now 23 locations. Then we run it through a capillary. And gosh, I had a capillary, and now I have to find it. But we run it through a capillary, which is a thin copper wire that is subjected to an electronic field. What do you know? I have a capillary. These are part of this, $200,000 device in the lab, but this is the real heart of it. You can't see it well, I realize that, but there is you can see the reflection of these copper wires. So this has 4 capillaries on it. The DNA starts out with these needles. It goes into the sample tray, and it sucks up DNA, and then the DNA races toward this window where a laser will hit, and we will detect the pieces of DNA. The smaller pieces make it through this tube quicker than the big pieces. There's a polymer inside, little tiny hole there, a polymer called POP4 or one of these similar polymers. And the DNA tries to race through it toward a positive charge at this end. Why? Because DNA has a negative charge over here. It always has a negative charge in water, and it's trying to race to this positive. The smaller pieces get there first. I run some known size fragments with it, and I can tell how big those short tandem repeats that I copied were. And I'll only see the things I copied. The rest of the DNA is too little of it for this machine to register, only the copied pieces. I have to interpret the results. I want to know how many contributors, and also from the quant, how much is male, how degraded it is, etc. If I find a match to someone, I have to apply a statistic. I'm now required to do that. It used to be we'd walk into court and we'd say, oh yeah, this pattern's consistent with this suspect, which could mean anything. It could just mean it's not dog DNA, it's human, and the suspect's human. That's all we could tell, so it's consistent. Again, remember Galileo said, count what is countable. And in 2010, the governing body over the FBI realized that, yes, you have to count what is countable. You actually have to give a statistic for the match. They are problematic, and we'll talk about them. Most labs now are using a probabilistic genotyping program when there's a mixture, more than one contributor. And I think that's an improvement, but we still see problems now. Every new medicine has side effects. And so there are problems with StarMix and with TrueAllele. We have StarMix in the lab, but I've used TrueAllele with the founder of that company. We have freed, I think, more than a dozen now people from wrongful conviction using this new statistical package that analyzes complex DNA mixtures. And then the report wording, which hopefully we'll get some time to talk about, that can be very, very confusing. And in fact, I think fraudulent. Testimony and cross-examination and closing statements. You, my friends in the defense community, need to be proactive about language. If the report says it's inconclusive, and there's going to be an expert up there talking about the DNA results, you need an agreement ahead of time about what inconclusive means. I suggest it means inconclusive. It doesn't mean, well, according to our technical details, I couldn't say he was in there, even though there was some things, I'm not saying he was in there, but there were some of his, none of that. If it's inconclusive, it's inconclusive. There are reasons for it being inconclusive. It may not be inconclusive in another lab that tests it, that has validated looking at more complex, very low levels of DNA. But if the lab itself says it's inconclusive, you want an agreement before trial that we're just going to say it's inconclusive. Okay. And then the language is important because the prosecutor in closing and the defense in closing will use language. And you want agreements about that language before you go. Are you allowed to call it a sperm stain if they never identified a sperm cell in it? Yes, they do that all the time, but they shouldn't. They'll call it a semen fraction, a semen stain, even though they've only done a screening test for semen, or they saw it fluoresce. There are a whole lot of false positives. And, there are a lot of reasons why someone might have a swollen abdomen. It's consistent with pregnancy, it doesn't mean that it's necessarily pregnancy. So you want to have confirmatory tests, know what that means. And if there's any conflict in testing, you remember those paradoxes that science doesn't like, you have to point those out. Okay, because that will be used throughout the closing and throughout the trial, that language, if it's called a semen stain, because somebody saw some evidence of semen, even though there was contradictory evidence, you need to head that off at the pass. If you were here, I would ask you about errors and what you saw happen. But you can write to me and tell me, or we can talk. Okay, so here's an alternate light source. It's a underwear from a case, and this is a great screening test. You want to know where to sample, but that's all this tells you. It doesn't tell you it's semen. Yes, semen has a characteristic glow, but we're not trained to tell that glow from other things. No one tests you on that ability. And so you need to do actual chemical tests that have been developed that can discriminate semen from feces, from urine and saliva, from dyes, from stains. Here's another example. This is semen at different amounts in a different volume. These are microliters, that's UL, so 5 microliters. 10 microliters is like a small teardrop. 20 microliters is a little bit larger teardrop. And you just see that, you can see if this is from the same concentration that the amount gets bigger and bigger. But again, this is not a confirmatory test for semen. This tells you where to sample, and then you can run some serological tests on it for proteins, or you can look for sperm cells. So you need to see all of these. Some states do not take photos of this. I just got to speak in Florida to members of 20-something labs across the United States. Some of them take pictures of all this and give it to defense, and others say, no, we don't do it. Like, so much can go wrong with a photograph. I got a photograph because I parked in the wrong space downtown. I was going to fight it maybe, but they had a timestamp on there. It's my car. And they took a photo for a $25 fine. This is a 25-year-to-life case, and we deserve justice deserves a photo because it's a subjective test. So how do we read these? Well, it's like the old-fashioned pregnancy kits. You put a drop of water that you'd put onto the underwear down here. If the water wicks up to the top, this is the C control line. It just means the water, under this plastic cover, you put the water down here on the paper and that made it up to the paper, this will turn red. So this is after the test is run. This is a positive control of the lab. They know they have control semen in the lab. This is a negative test. So this is a P30 test. That's one of the common tests. It's called prostate specific antigen, sometimes PSA. And it will light up with a band, or it'll give you a different color band because of monoclonal antibodies right here in the middle at the test line if there's semen proteins detected. Now, this has some false positives, but it's a really good screening test for semen. And here's the negative control. You'll notice the water made it up. They just put pure water, made it up to this line. We get a control line, no test. All right, so here's an actual, a different one. This is an acid phosphate test, another protein that's elevated in semen, but you can get false positives with a number of other body fluids or body substances. But anyway, it's a good screening test. Control positive, you let it incubate for a while, and this one, maybe there's a little bit of pink in there. And this is why the labs are a little nervous about supplying photos. It's hard to tell exactly. It's not the same as what you see, but that's why you run the positive, negative control, strong ones, positive and negative. All right, so let's look at some actual evidence now that you know how to read these. Ready? So is item 4 positive? So here's a test. That's the positive control. See how brightly red the little bit of substrate that's put in there turns. Here's the negative control, a little bit of pink. You leave these out, the color tends to enrich even in the negative after a while. But here is item 4 run through several dilutions, and here is the substrate control, which just means part of the underwear knots or the leggings, I can't remember what this was in this case, not stained part. And you can see the not stained part's kind of pink, it kind of looks like one of the diluted samples, but even at strongest, does that look like this? Well, that's what we need to see. We need to talk about this. We need to know, were there other tests? Was there a P30 test that conflicted with this or confirmed it? So let's look at P30, another semen test, another protein enriched in semen, prostate antigen, and let's look at it. So here is that cassette again, and here is a test for item 4. And do you see from item 4 the different stains from item 4? And do you see on item 4 any positives? Well, here we see that 1 through 3 were negative, according to lab, but 4 was a very weak positive. It was. This is a positive, right? This is a negative. Now, I believe them that they saw something here live. There's no question. But we need to have an objective way to corroborate or refute something that is subjective. And this test doesn't show me a clear answer. But you wouldn't know that for many states. I was supposed to fly to one state last week, to Minnesota, because they don't take pictures at their lab. And I had, I've talked to people, and they think that, well, they're experts and they can call this. And I'm like, yeah, but how do I'm an expert too, but I don't know what you saw. I need to see what you saw because it's subjective. Okay. So words matter. Here's a semen identification report. And what we need to pay attention to is, how does this impact your trial? So results and conclusion, they want to tell, was it a semen stain? If there's a semen stain in the child's underwear, that's very serious evidence. And so a visual and light source for the panties revealed stains characteristic of semen. And results from chemical and serological testing of the stains in the panties are consistent with seminal fluid. Really? Consistent with, remember that. How about a number for this? How about, what do you mean by consistent? This looks paradoxical to me. It doesn't look like a real positive result. In fact, you didn't see any sperm cells. That would be consistent with positive, but you got a negative for sperm cells. How do I know that? Because it says microscopic examination, that means looking for cells, from smears prepared from the stains, fails to reveal the presence of identifiable spermatozoa, sperm cells. Okay, what happens to this item? Well, this sounds like it sounds like they think they found it, but they did say weak positive when we went to their bench notes, and look at the photograph. Okay, what's that going to do for the case? Well, I'll deal with it during trial. During trial, this is called the semen stain from the child's panties throughout the trial. Closing arguments all about the semen stain on the child's panties. And the semen stain revealed the identity of the accused. What does that actually mean scientifically? It means that the accused, there was DNA from the underwear or the leggings that is consistent with, and maybe a really strong match with, the accused. But the accused says, hey, child was on my lap, they use water, it soaks through the underwear. They could have picked up DNA from the outside, or I fold the laundry, or we do laundry together, they pulled this out of the laundry, or whatever. My DNA is not unexpected on the underclothing of children that I care for. My semen, on the other hand, I mean, it's possible to get mixed up in the laundry or whatever, but my semen is, far more probative. But we can't tell, first of all, from this, was it semen? And second of all, whose semen it was. Because the proteins that indicate semen or not, or the visual examination, are separate from the DNA test, which is just a DNA test of a piece of cloth. Now, you can use your inference, and you should, if there's a big stain. And in this case, the lab did a great thing. They took a bit from outside the stain to show a substrate control. And if that shows a strong profile and a strong reaction for semen and maybe a lot of sperm cells, and it's the only place where the DNA from this male person is, well, you use your inference for that. But the science cannot tell you whose blood is mixed with the DNA or whose sperm cells they were. There are methods to do that. None of the crime labs do it. None of the crime labs cut out the little sperm cell one at a time and test them. So beware of this type of language. Our words matter. And to the degree that you allow that to happen, you better be sure that the damning words that are used are accurately used. And your keys are, before trial, an agreement about what words should be used, objections to every misuse of terminology so that it's in the record, and attempts to get the labs to stop doing this stuff. I know that that's not your chief job, but that is your effect. Okay, so here's the Christmas tree stain of sperm cells. Very beautiful. Oftentimes you'll get some green in there too. We see the shape of the sperm cell. It has this characteristic kind of eyelid staining that happens. The tail, and then of course this junction here that is a little bit thicker near the tail, stains up pretty well. And again, these are tough cells, so we can break all the other cells in the mixture and just leave, try to leave just this. So here's how it actually, that was a textbook picture. Here's a case I worked on, police officer on a campus accused of coercing a young woman to give him oral sex. The investigator interviewer notices she's wiping her mouth on her sleeve when they're talking, and so he says, "Can I have your jacket?" And he asked the lab to test the jacket, looking for traces of semen, sperm, the officer's DNA. And the lab does that, says, "No, we didn't detect anything." And he's like, "Swab it again, please." And that happens, and you see that in the emails. And so they swabbed it again. And in that case, they got a sperm cell, they said. So they took a picture, and that's the picture we got. And well, it has some characteristics of a sperm cell, but this is an outdoor jacket. It could be, there's pollens, there's all kinds of things that might pick up the stain. Here's some junk. I took a picture of the junk, and I took a picture of the sperm cell. And, you know, there are 2,800 of these possible fields of vision. This is the one that they took a picture of where they saw a sperm cell. I don't know how many they looked at where they claimed to see a sperm cell, but I thought it was important for the jury to see this. And so I made a slide. And also it's important to say, in that second swabbing of the jacket, it had amylase, which you get from your saliva. Well, she's wiping her. Do we know whose amylase it is? You know, I mean, she's wiping, and they detect amylase, which is enriched in saliva, a protein. But it was acid phosphate negative, which is enriched in semen. So you have a negative result for semen. They have at least 5 profiles, DNA profiles of people on there, probably many more, but at least 5 that can be discerned in an S1 STR test. And at least 3 of the contributors are male, putting DNA on there. However, because the lab attempted to get an enrichment for sperm through that method I told you, you differential extraction, you break open the epithelial cells and you hope to have enrichment. Oftentimes that second fraction has a lot of the female DNA in it, just because you can't wash it all off. And just because you tried to get sperm out of it, if you don't see sperm, if you don't take a nice picture, why should I believe you? But the problem is, many labs still refer to this, most labs still refer to this second extraction, this attempt to enrich for semen, as a sperm fraction. Now, they can call it a sperm fraction even if they see no sperm in it, even if they have contradictory evidence about there being even semen from the area, from the sample. I brought this up in my meeting a couple of weeks ago with 22 labs down in Florida, and there were a couple labs that say, yeah, we dropped that terminology. We just call it the second fraction. I said, well, why did you do that? Can you explain to everybody else? And they said, because we would put our experts on the stand calling this a sperm fraction, and there would be only female DNA in it. And we know that that can happen in the lab. That is an attempt to get sperm. It doesn't mean you're going to get sperm. Sometimes all you get is more victim DNA. They said it was confusing to the jury, and it was confusing to the officers. The police officers were like, what do you mean? It's a sperm fraction. It doesn't have any sperm. And I came up with all kinds of great analogies about like selling a gold fraction in the marketplace. And it's like, yeah, but is there gold in it? Well, it's the gold fraction. Yeah, but is there gold in it? You know, oh, it's just a marketing technique. You wouldn't swear to that in court. Yeah, they swear to it in court. And the effect, the absolute insidious effect, it is absolutely medieval in its poor legitimacy. The effect is that this DNA is called throughout the trial the sperm fraction DNA, even though the amounts found don't really justify calling it a sperm fraction. That the one sperm cell that they saw from, or that they think they saw from swabbing all this, doesn't let me legitimately think of this as sperm. It doesn't matter. They don't call it the second fraction. They call it the sperm fraction. They'll call it the sperm fraction even if it's mostly female DNA. Even if it's only female DNA, they will still call it the sperm fraction. Some labs have gone away from that. You need to be aware of that. Okay. There is some studies on this differential extraction. When can you legitimately say, yeah, it's sperm? Well, if you see sperm in it, you just take a sample, put it under a microscope, you can call it the sperm fraction. It might still have female DNA, some female DNA in it, but you can call it the sperm fraction. Also, if you get to a certain ratio of male to female DNA, a certain amount of male DNA in that second test, there's one learned paper on it that says that you can call it a sperm fraction. At least in this lab, they were able to show. But there are low levels of male DNA where they got false positives for male, that they were calling sperm fraction something that they know they only put female DNA in. And that's the kind of research that you have to do, and not all of your DNA experts are going to do that. And there's, I'm sure there's some stuff I miss. This just is a really careful reasoning through that a common person, if you're told it's a sperm fraction and they say, but there was no sperm in it, that should be an oxymoron, right? A paradox that is not science. This is, the size of the things we're using to transfer DNA, a pipetter, which is just a kind of an eyedropper or a turkey baster, and but for transferring small volumes. And we're working with tubes this close, and those tubes can get mixed up, or you can have a little bit of aspirated DNA that gets into the air that might, or in an instrument that might contaminate from one tube to another. Remember that when we get these samples, we're going to copy them with polymerase chain reaction in the lab. What does that do? It takes that one cell's worth of DNA and turns it into a billion cells or hundreds of millions worth of DNA. Because remember, we're copying to measure that stuff. That means if one cell from a suspect gets transferred into a tube with evidence, from then on, that sample will have that evidence DNA in it, because it will be made into a billion copies, and some of those copies will come out anytime I go into that tube. This is the machine. It's about the size of a large oven. I showed you the capillary part of it in our show and share. And then this is the electropharogram that you will see if you properly request the discovery from the lab. Let's look at one of those locations. And anytime you see a graph, you want to know what's on the x-axis, the horizontal line. And here it's base pairs. At the top, you see 80, 120. Those are those AGCTs. They're the number of bases in length. But the computer's smarter than us. And so it will say, hey, I know how long the piece is. I know how long the primers are, how long the area is I'm measuring. And what you care about is how many repeats there are, short tandem repeats. So I'll put that in a little box here, this 16 and 17. So this is the D3 locus, DNA locus on chromosome 3. Some of them have really good names, tell you where they're from. And there's some other numbers after this. I'm just going to shorten it to D3. And we see the person got a 16 from one parent, a 17 from another. Remember, the last step is that statistics. And so we go to a database that the FBI was nice enough to put together. Let's go to the Caucasian database. So for white people, they had an N of 203. That means 203 people gave their DNA to this. So up to 406 chromosomes. And sorry, for each allele we're measuring, everybody gave a pair of chromosomes. And here are the alleles and what percentage of the population as a decimal is each of these. So let's look it up. 16. Why 22% of the alleles from Caucasian people, these 203 Caucasian people, are 16. Coincidentally, totally coincidentally, 22% are also 17. Because they're two different sizes, we multiply by 2. If it was just one size, like the next one is just that 15, we just multiply the frequency of the 15 times itself, or square the frequency. But because there's two of them, we multiply by 2. I don't expect you to do complicated math, but if you multiply 22% times 22% times 2, you get 10%. 10% of the Caucasians listening to this, if there's a sufficient number, we would expect to be a 16, 17 at D3. But you do that for all these sites, and you end up with 1 in 80 quadrillion, because you can multiply. These are independent lotteries you're winning for this match. So you multiply those different percentages, and you end up with a tiny number. How much less than 1? Well, just divide 1 into 79 quadrillion, or 79 quadrillion pieces, and one of those pieces is what we're talking about. That's how unlikely it is that we would find another person who isn't an identical sibling or related to this person who would be this. Now, that database and the statistics we produced are for unrelated people from the proper racial group, unrelated people chosen at random. What if there's a relative of this person who might be a suspect or might have left DNA on underwear or a gun or something? Then these numbers fly out the window. They have another set of calculations, and it can be drastically lower. Okay, so I showed you how small those tubes are. Why is that important? Because people make mistakes. It's like asking for ketchup at the drive-thru. You might not get ketchup. You might get mustard. You might get both. You might get whatever. And these are humans doing this, though they'll tell you robots do it. Well, robots do part of it, but the people still bring out the little racks of tubes and set things up and take stuff out. And so here they had the evidence and the suspect. These charts refer to where the tubes or the wells, sometimes these are there's no cover on these. They just put the liquid into a tray like this. This tells you where they were, and it tells you that they were on the same tray, which means they had the evidence out at the same time as they had the mouth swab from your client. And they're going to tell you, we were very careful, but there's no reason to do this. You can do all of the evidence first, and that's what we prefer. That's, I don't want my son's DNA out with evidence from a rape or a murder, right? I don't want my daughter's DNA out with any kind of evidence. I'd say do the evidence first and then bring their DNA in. And that's what the best labs do, but most labs have exceptions of that, and oftentimes you'll see this. This needs to be challenged and fought and stopped. So anytime you see it, please do. This is that quantification. You need to see this. This is all in the bench notes. These should be in the bench notes that you asked for. And this shows the male to female ratio of DNA and how much degradation. It's a product called Quantifier Trio. Most of the labs are using this. So, here are the fingernail scrapings from the actual case. We have the concentration of human DNA, the male DNA, and then they give you this nice one, the percent. What percent male is the fingernail? Well, it's less than 1%, but that's okay. That might be enough to test, and we can do that calculation. You just want to know, though, how much male. And there's much more DNA off of these fingernails, right? So you can get sometimes a lot more from similar samples. And then I asked for some other things in this case. So I wanted to know how much total DNA is left, because if there is 0.1 nanogram, as there is here, that's enough to test. Here there's 0.8, that's certainly enough to test. And so I wanted to know how much of it's male, and that's what you get from your quant. You get the concentration of male, from which you can figure out, is there enough male DNA to test if they concentrate. And that degradation index, sorry, that's on here as well. This is the small fragment, the 80 base pair one versus the 240 base pair one. How much better does the 80 base pair one copy? And that tells you how degraded it is, because the big pieces break up when it's degraded. And so in this case, the little pieces are 4 times better than the big pieces. That's okay, we can deal with that. I won't go through this, I don't think we need to. So here's an actual electropharogram from the Amanda Knox case. This was a knife taken from the apartment where a man that Amanda was dating lived. The victim, who was killed with at least one knife, probably one knife, the victim was cut very severely, also cut a few times before or around the time of death. And this knife was a kitchen knife, a rented knife in a rented apartment that this young man who Amanda had been seeing for a week or so, a few weeks maybe. Anyway, from his place, the victim had never been there. Amanda's on the handle. She says, I slept there that night, I cooked. Yeah, my DNA is expected. It's like the top knife in the drawer. We don't know why the police collected just this knife out of all the knives, but they did. And they tested in 4 places on the blade. They found Amanda on the handle. Nobody's arguing that. But on one of the places they found this DNA, there's that locus D3, its full name, D3S1358. We looked at that. And it gives us the repeat number here, and also how high the peaks are on the Y. Now that means how much DNA copied, that Y axis, how tall it is, how much DNA copied. And so this is 47 units. They call them relative fluorescent units because we add a fluorescent dye to the DNA. This peak is a little less at 18, but this is consistent with the victim. So it looks like that should be good evidence for the state. Why would we say this isn't good evidence? Well, these numbers here tell us how big that peak is. And if you remember the earlier one, the peaks here are 1,000, 1,500, 600, but the peaks in the one piece of incriminating DNA in Knox's trial, the peaks don't go, only one of them I think is above 100. And as our report reflected, and the DNA experts that Amanda had at trial with her tried to explain at her original trial where she was convicted, this is not a reliable piece of evidence, because none of the other, none of the labs in America anyway, the ones that I was studying at the time, would ever look at anything this low to confirm guilt. The FBI wouldn't look below 250 units. And here, the highest ones are 100 or so, and most are below 50. My own lab was looking at maybe down to 150 at this time. But, we knew that we couldn't reliably look at stuff this big. Why? Because sometimes there's contamination, because our instruments haven't been validated to that level. So why was this one lab, which was going through it's oh my gosh, what do you call it when a lab gets approved? Anyway, they were going through the process of being an accredited lab, but they didn't supply any documents that let me think that they could do this where nobody else was doing it. So that's the kind of scrutiny you use. You have to look at the lab, validate what it's measuring. Remember Galileo, count what is countable, measure what is measurable. One of the things you have to measure is how good you are. What is your error? When do your methods fail? And that's what we look for in those validation summaries. How little semen are they no longer able to detect? Where does their test sometimes give them a positive where they know that they didn't put any semen in that sample during validation? So we look for where validation failed. A lab that has no validation study is invalid. Okay, so what about these tubes I keep talking about? Well, the order of operations is important. Does it, will you get every case thrown out where they don't use the best practices of doing all of the evidence before they even bring the suspect DNA in? No, you will not. But should people know the care or lack of care that a lab is using and the risk of contamination? Well, you need to request from the lab all of their examples of contamination for at least 2 years before your case and 2 years after, if it's been that long. And you'll see that there's contamination in every lab. That is, sometimes a worker's DNA ends up in a control tube. They can detect that. They can't detect this type of contamination where a suspect's DNA accidentally ends up in evidence. So that order of operations, just like washing your hands, you wash your hands after you use the toilet. Why? I don't care if there's grease from cooking hamburgers on the handles of the wash stand. What I care about is there feces in my hamburger and from under this person's nails or on their fingers? And so wash your hands after. Do the evidence first, the suspect after. I showed you that plate. And am I just being fussy about these little tubes and like, hey, these people are careful? Well, this is a case from Las Vegas where the Las Vegas lab had 2 suspects in a kidnapping and robbery of a woman and her child. And the lawyer, as you lawyers have to do, had to advise the client of a deal they were making. Client, they have your DNA. It puts you there. It puts you on, her belongings, and it matches you. We can go to trial. You'll be facing whatever 25 years, I don't know what it was. Or you can plead. They're offering an agreement, and we're hoping the prosecutor will be lenient, maybe give you 12. You'll do 8 or 10. What would the client do? The client took the deal after 4 years, I think, in prison. California called Las Vegas, the California lab, and said, we have a match to that sample, that forensic sample, you loaded up. And Vegas said, no, you don't. The guy's in prison. He's been there for 4 years. We have him. It's a match. We just did it. Well, you're wrong. No, you're wrong. No, you're wrong. No, well, let's both swab. So they both go back and swab the people. What happened? The 2 tubes from the Vegas case where both of these men, the man in California who'd been in the system, put in the system, and the man from Vegas who was imprisoned, they were testing those samples at the same time, and they mixed the tubes up, or they mislabeled, or they mistyped, and so they apologized. Okay, we're at the end. As you can see, there are plenty of slides left. I'll just show you one last one to tell you that this problem of scientific bias is not going away. You must read this paper. This is Ideal Drawer's paper, February of 2021. And he asked forensic pathologists, he gave them the exact same data from a child who died. And these people are medical doctors, special training in forensics, testified about cause of death. Is it accident, suicide, or indeterminate? Accident, sorry, accident, some sort of homicide or indeterminate. And the only difference was he told half of the people the following story. It is a white child being cared for by their maternal grandmother. And in the other one, it is a black child being cared for by the mother's boyfriend. So the black ones are in the black bar, the story of the black unrelated male, the white related female. And you can see that of those who could determine accident, there were 26% of the ones talking about the grandmother thought it was an accident, and I don't know, 7 or 8% with the same exact medical information said it was the accident for the boyfriend. And the more important thing from a defense lawyer's point of view was that the homicide of the unrelated black male, these are experts. These are the people who tell the police if it was a homicide. Those experts took the same evidence, and 35% of them decided it was homicide with that same evidence when it was the black unrelated male, and 12% when it was the white grandmother. Now, this is 2021. This received more letters at the Journal of Forensic Sciences than any article ever. There is a great, look it up. There's 12 letters that I think they published. The editor offers an exasperated editorial. Ideal's a colleague of mine. We wrote on DNA bias and mixture interpretations, and he had to answer all of those 12 letters. He's being hounded by these professional groups that have called for a retraction of this paper, threatened all kinds of things. And this is going to happen because this is us, not because these are bad people, because human minds must be constrained by systems and by lawyers like you who have agreements about what language is and who scrutinize the evidence and launch objections and try to work with prosecutors to stop these practices that do not allow the triers of fact to make the correct decision. So thank you very much for your time. You can easily get in touch with me. I'm quite easy to find. If you have questions, please get in touch. If you have cases that I need to look at, get in touch. Let me know how I can help you promote justice. And I think my final slide was my contact information. So I'm just racing through all the rest of this. If you have questions about the other slides, let me know. I'd be happy to talk to you about them. There's an awful lot of them. Thanks very much. Whoops. And there's the last slide. If you want to get in contact with me, the best address is forensic DNA analysis. It's at gmail.com. That's wrong. Forensic DNA analysis at gmail.com. And I'll just end it with that. Thanks very much for your attention. Let me know how I can help you.